Uh, Good afternoon, everyone, and thanks for joining us for the CBAM seminar today. Our speaker today is the director of CBAM, Dr. Vaughn Cooper. Vaughn received his bachelor's degree from Amherst College and then completed his PhD in the lab of Richard Lenski at Michigan State University, where he studied the evolution of specialization in the long-term evolution experiment of E. coli populations. From there, he joined the Society of Fellows at the University of Michigan and studied the evolution of various bacterial pathogens of the cystic fibrosis airway with John LaPuma. Vaughn started his independent research career at the University of New Hampshire, where he earned an Outstanding Assistant Professor Award as well as an NSF Career Award. He moved here to Pitt in 2015, and his lab is focused on understanding how pathogen populations evolve to adapt to new hosts and new environments, both in vitro as well as in vivo. So one of the things that I really admire about Vaughn is his ability to see a need and then figure out a way to meet that need. And I'll give you 3 examples. First, Vaughn saw that most high school curriculum for teaching evolution is really lacking, and so he developed an inquiry-based lab approach to understanding evolution called Evolving STEM. Second, Vaughn saw that researchers like me needed a quick and efficient way to sequence bacterial genomes, and so he started the Microbial Genome Sequencing Center to provide researchers with high-quality sequence data both quickly and cheaply. And finally, over the last year, Vaughn has found multiple ways to meet the various needs presented by the COVID-19 pandemic, from offering SARS-CoV-2 sequencing services through MIGS to posting YouTube videos about the evolution of the virus. And so we're really delighted that he's agreed to take us on a deep dive into SARS-CoV-2 evolution today. So without further ado, I'll stop there and hand the floor over to Vaughn.
Wow, thanks, Daria. There's nothing more flattering than such a warm introduction from one of your sort of most valued colleagues. So I really appreciate that very much. I will start screen sharing. And we are good. So thanks everyone for joining, especially all of you who found the link through Twitter or wherever else. And thanks to everybody from our local community. You know who I am now. I'm Vaughn Cooper, and I'm in microbiology and molecular genetics in the med school. I am thrilled to be kicking off our 2021 virtual seminar series for the Center for Evolutionary Biology and Medicine. CBAM's aim is to catalyze research and education in evolutionary biology that can improve medicine. We really encourage you to stick with us and join us. One of our major goals in the center is to share how evolution is important for medicine, and this is especially true for this ongoing COVID pandemic. And I really want to recognize the CBAM Executive Committee, Matt Nikotra, Associate Director, Ann Carvunas, Dennis Koska, Eric Wright, and Daria Van Tine for putting me up to this challenge. I also really want to salute and thank my amazing research lab and collaborators who've supported my endeavor into studying this virus over the past year. It's not funded, it doesn't really overlap with much of their work, and it's definitely taken my attention from many of their projects at times, but I'm really grateful for their inspiration and feedback and their patience. So thank you guys. So at the start of this pandemic, the Atlantic magazine actually recalled, not remembered, but actually said, hey, come back, the superstar science journalist Ed Yong from book leave. And they asked him to report on this pandemic, but not just to report on it just as a reporter, but really to take a big swing at the core issues we're all facing. And if you've been following any of his writing, you know that he's succeeded in spades. In fact, he's taken many enormous swings that have really illuminated my understanding of what's going on. But I heard him say this in an interview and I thought, wow, you know, I'm an evolutionary biologist who studies how pathogens evolve. I've essentially been training for this moment for 25 years. I really wish I could do that. I was thinking this on a bike ride. And then I thought, look, I'm a tenured professor. What excuse do I have? And so after a year, this is my first swing. And the last thing I want to do is be compared to Ed. That's a fool's errand. But now that I can finally start to reflect a year in and wonder what actually have we learned about evolution, I thought it was time to share. And so another of our great science writers, the leading writer on evolution, I think, Carl Zimmer, wrote this tweet to introduce his coverage of our article that I'll discuss later in this talk. And he said that pandemic is like course in evolutionary biology. And I'd also been thinking this, sort of, which leads to these questions. What has evolutionary biology taught us about SARS-CoV-2? And what in turn has SARS-CoV-2 taught us about evolutionary biology? And so the first question, an evolutionary question, is where this virus come from? Like, what let the cat out of the proverbial cat out of the bag and caused this pandemic? And to first explain, let's consider a family tree of cats, because who doesn't love cats, right? And it's also really key that we get a good handle on understanding phylogenies, because it's key to understanding this virus. So what I'm showing you here is a family tree of cats as an example. And this is a complete sort of evolutionary family tree, or phylogeny, which is a scientific model of relatedness based on the complete genomes of these different species. And this figure indicates a series of nested groupings among cat species. And at the far left, I hope you can see that the lineage that gave rise to cats diverged from the common house cat lineage about 12 million years ago. Now, over on the right, you can see that the snow leopard and the tiger are actually sister species that diverged from one another around 3.5 million years ago, meaning that snow leopards are more closely related to tigers than they are to leopards, which I think is pretty cool. So in any case, we can use the same rationale to infer genetic relatedness, relationships between different kinds of viruses isolated from different host animals. So now I'm showing you a family tree of coronaviruses. And that's the one, it includes our newest one that we know about, SARS-CoV-2. And the orange arrow is pointing to this virus. And together, this group up here at top is known as the severe acute respiratory syndrome associated coronaviruses. And now I'm highlighted all the coronaviruses known to cause human disease. And at the bottom, there are several coronaviruses down here that cause a common cold, and their closest viral relatives are rodents, mice and rats. But above are the other coronaviruses that cause more severe diseases and have caused major epidemics in the last 20 years. MERS, the original SARS-1, and now SARS-2. And all of these are closely related to viruses isolated from bats. And more recent analyses have expanded the number of close relatives of SARS-CoV-2 to isolates from bats, specifically in Cambodia and China. So all of the severe coronaviruses trace the recent ancestry to bats, but it's possible they pass through another animal like a pangolin or a civet immediately prior to infecting humans. So sort of raises the question that you've probably thought about: what makes bats so special as hosts of viruses that cause human epidemics? And the first explanation is that there actually just are a lot of bats. So 20% of all mammalian species are bats. And that makes them a pretty sizable and mobile reservoir of viruses that can infect other mammals. The second is that bats have evolved a broad range of metabolic capacities that enable their success as fast-flying predators and their relative long lives for their size. They can live several decades, and they have exceptional defenses against byproducts of their high metabolism. And the last is they actually have fairly specific antiviral responses, ironically, through one pathway known as STING. And that STING pathway is actually dampened. So their immune responses to viral infections are actually more measured or modulated relative to ours, and thus their responses are not as damaging to the bat. So they tend to tolerate and manage viral infections better than we can. Now, a very new discovery by Stephen Goldstein, a new collaborator of ours, working with Nels Elde at Utah, found that SARS-CoV-2 or SARS-like CoVs are prone to genetic recombination with related and even distant coronaviruses in regions that are important for host tropism, which host they provide or preference. And this figure from their new preprint that's on bioRxiv demonstrates this using a new bit of software that they've developed. And what this software does is indicate regions of exceptional dissimilarity that are unlikely to be explained by stepwise evolution alone. And you'll note, hopefully, that there's a big peak of recombination, inferred recombination events here in the spike protein. So it appears that CoV have undergone recombination in critical regions for binding and uptake. Now, you've also probably heard many reports questioning the origin of SARS-CoV-2. And so just to put some numbers on this, the closest relative in this more detailed phylogeny provided by the invaluable sequence database known as GISAID is about it's 96% similar to the original SARS-CoV-2 genome, which means that based on the rates of evolution, which we're going to cover soon, and at the current mutation rate, which we'll talk about, they're about 46 years diverged from one another based on the 1,110 mutations that differentiate this most closely related isolate and original SARS-CoV-2. So of course, recombination events like the one I just talked about could shorten that time, but the SARS-CoV-2 is actually fairly distant. So it doesn't appear that this at least is a direct relative from one of the stocks from the bats isolated in Yunnan province. Okay, so we know that this novel coronavirus came from bats. We knew there were many potentially dangerous bat coronaviruses based on the previous two pandemics, SARS-1 and MERS. Their capacity for recombination and their capacity for various forms of pathogenesis is even clearer, and I won't get into the pathogenesis mechanisms in this talk. How is this virus now evolving in humans today? So the evolutionary changes with the novel coronavirus have been captured and analyzed really beautifully by the group who built and run nextstrain.org, and that's Trevor Bedford, Rickard Nair, and many others, including Emma Hogcroft, who I'll be talking about more, whose work I'll be talking about more later. So Nextstrain not only depicts changes in SARS-CoV-2, it also tracks other epidemic viruses like influenza, Ebola, and others, including some of the endemic coronaviruses. And on this graph that was a snapshot from GISAID just a couple of days ago, the x-axis is time. And over the 15 months of this pandemic, genetic diversity has mostly, has arisen by random mutations, and we can plot their accumulation and their spread over time. And on the y-axis, this is the number of mutation differences out of the almost 30 kilobase or 30,000 nucleotide genome. And this particular plot is based on a subsampling of the nearly 800,000 genomes there are, actually more than 800,000 genomes there are now available. It's amazing, 800,000 viral genomes. Just pause and think about that. So this is vastly subsampled, but it's fairly representative. And from this data, we can infer that the average distance of any isolate on the planet right now is about 30 mutations different from the original Wuhan 1 isolate way back down here at the lower left corner. And that original isolate is almost certainly extinct. But because all of these lineages trace back directly to that original isolate, and their reads, if we sequence them, can map directly to that original isolate, and we can verify genotypes quite efficiently by that mapping system, I would say this is still just one strain. So another view of this same data is with these radial, unrooted radial phylogenetic trees. And I hope you can see there's pretty notable lineage structure here. There have been at least 12 major lineages that have evolved around the world, and these are mostly distinguished by random and inconsequential differences. And the viruses from the same lineage are certainly much more closely related to ones than those of different lineages, but those differences do not usually define fitness differences or adaptive differences. Rather, these differences are extremely useful for genetic or genomic epidemiology. And some lineages are actually more common in other regions. So at the left, this is actually the global sampling, and at the right, this is the North America focused sampling. And hopefully you can see that this yellow clade, known as 20G, is much more common in the United States than it is worldwide. So the US population can be distinguished somewhat from global samples, of course, even though we're clearly interconnected. And so as I noted, these lineages do not at face value define adaptations, but we can query these data and ask, are there particular mutations that are characteristic of potential adaptations? And so what I've done is queried this database for genomes with the 2 amino acid deletion and spike that was found in the very well described now UK or B.1.1.7 lineage. And then I've done the same here for the US lineage. And hopefully you can see that, first of all, this is actually a bigger slice of the global pie than the United States. Not by that much anymore, but it is still more significant. And hopefully you can also notice the shape of this phylogeny. So it arose newer. Time is going from inside to out. So this is a newer clade. And that newer clade has expanded and diversified somewhat a lot recently. Same structure is true here, although that pattern of diversification in the US is a slightly more delayed because it sort of came from the United Kingdom or thereabouts over to the United States shortly thereafter. Okay, so that's the sort of broader landscape of mostly random variation in SARS-CoV-2, and we can again query that data. We'll talk about that coming up. But one truly underappreciated fact of viral evolution is that no matter how high the titer of an infection within a given infection, the key variable dictating their evolution is the bottleneck size. So a higher mutation rate will introduce more genetic drift, but the potential for adaptation by natural selection depends on the bottleneck size and the overall number of total infections. Now, if the bottleneck is large, selection will be stronger because it can distinguish among a larger pool of potentially adaptive or maladaptive variants. But if the bottleneck is small, selection is weak relative to drift. And so that brings up the key question: what have we learned about SARS-CoV-2? And I think it was pretty clear even from early on that the bottleneck was small, just based on the patterns of genomic epidemiology. But quite recently, a paper that had been at preprint for quite some time just came out, led by Katrina Lithgow with Tanya Golubchek's group and many others from the COVID-19 Genomics UK Consortium. And they figured from tracing whole population deep sequences of many infections with sort of known chains of infection, that the bottleneck size for SARS-CoV-2 is anywhere from 1 to 8 viruses, and it tends to be biased towards the lower number of that range. But by deep sequencing, you can trace whether there are minority variants, mutations that are of reaching intermediate frequencies within these patients, and the numbers of mutations within hosts basically on average was between 0 to 5. Some mutations of those 0 to 5, they sampled across many, actually recurred often. And so out here, this is representing a particular mutation that has occurred 17 independent times among this large data set of infections that they sampled. But most mutations remain quite rare. And so this is the number of sites in a sample. And what this is just saying is that in general, most mutations don't reach high prevalence. Okay, so take home number one, the bottleneck size is quite small and the amount of variation of any given infection is relatively low, which means the probability of a new mutation arising and being transmitted for any given infection is low. The average number is actually 0. So I want to highlight a collaboration from our center that we put together early in the pandemic with Eric Wright and Seema Lakdwala, a virologist and member of the center in my department. And what we did, and really what Eric did, was built a massive phylogenetic model of all of the viruses back then, and this was back in May. And we looked for clusters of related sequences that were indicative of transmission change. And then we asked if some occurred repeatedly, with the logic that repeated evolution might be a signature of beginning adaptations. And we found that some mutations repeatedly evolve and form clusters, such as the L5F mutation in spike, which is one of the very common mutations that the Lithgow et al. Identified. However, and we also noticed a very strong CDU bias. And so we were one of the first publications to show this strong CDU bias in the mutation spectrum of SARS-CoV-2. Why that happens is still uncertain. There's been some suggestion it's related to APOBEC-mediated DNA damage and DNA repair processes. But one thing that we noticed is that at that stage, in a way, some mutations were especially mutation poor. And so to explain what I mean by mutation poor, I want to share a quick story from World War II. And maybe that's appropriate because some experts have compared the pandemic to the experience of a world war. So a group of Navy engineers was studying where their bombers had been hit by enemy fire to learn how to improve their design. And so here's a schematic that summarizes all the places where bullet holes were found on the plains. And so the initial response is, oh, well, these are the regions that got hit a lot, and so we really need to invest in shielding those areas of the plain. And then, in fact, there are some areas of the plain that were no holes. And so the point of this lesson is actually the reason why there are no holes is those plains didn't make it back, right? So those are regions of the plain that sustained such severe damage that they were shot down. The ones where you see the bullet holes were actually regions where the plane was able to fly home. And so this is now known as kind of the case study of survivorship bias, coined by Abraham Wall. And so the key is we need to pay more attention to the survivors and not the missing. So we applied that same analysis to mutations in the coronavirus. And we found, of course, there's some genes that are hit often because they're big, but there are also some regions that were unusually mutation poor. These two mutations encode the RNA polymerases and their helper proteins. These are the targets of the antiviral drug remdesivir. And so this is just one example of how an evolutionary analysis can identify conserved regions as evolution is happening to help design potential therapeutics. Now, remdesivir hasn't been the hope that the salvation that we were hoping for, but it's still being used clinically. Okay, so at the beginning, right? So we mostly have random variation, not much in the way of adaptation, very small clusters of populations. So even though there were certainly some major flare-ups in cities like cities in Italy and cities in our country, like the United States, but it was mostly slowly and randomly diversifying as a population. But then the runaway pandemic that occurred when we mostly as a country basically gave up caused case numbers to explode. And this was like a runaway, like an evolutionary windstorm over smoldering embers with nascent variation. And so this is the effect, this is the trajectory of infections in the United States from March to June. And at this point, the effective population size, or basically the actual genetic diversity that selection can act upon in any given time. And then the pandemic exploded, as you all know, and that caused an effective population size of approximately, say, 50 odd different viruses being acted on by natural selection to double or even go up by 100-fold, depending on different estimates. And so with that huge increase in effective population size, natural selection starts to gain the upper hand. And so the first example of this is the mutation called in spike called D614, or known as D614G. Some have called it DUG for D to G. This initial mutation spread very rapidly throughout the world, and its spread actually met a lot of circumspection from people like me, thinking that this could be explained simply by sort of demography or luck. But later, many careful studies showed that it was a combination of demography and luck and a moderate and measurable fitness advantage that improved its growth rate relative to its ancestral type. And now more than 90% of all viruses on the planet have the D614 genotype. So this is basically the first major selective sweep. And in fact, there's actually another variant, D614N, that's found in the remaining sort of 5 to 10%. So mutations at position 614 have evolved. Then another thing that started to happen is, frankly, with more numbers, more infections of immune compromised individuals. And so it's long been known that immunodeficient individuals are susceptible to chronic infections by viruses, bacteria, and fungi. And that can enable larger pathogen populations persisting over longer time periods. So whereas a healthy individual may have strong immune control and surveillance that sort of censors pathogen diversity within that individual, an immunodeficient individual will be infected for longer periods of time. That pathogen population will be larger and have essentially more evolutionary leeway. And so I've got this backdrop of actually spaghetti stuck to a wall. And this was suggested, these are great words from Elaine Vitone, who put together a really fun Pitt Medcast podcast about some of our work. And she said that what happens during these infections is like the pathogen is throwing spaghetti at the wall, and most of that diversity is useless, but sometimes a good mutation sticks and is even able to transmit to a new individual. So you get the idea: more cases, more instances of chronic infections, and more opportunities for genetic diversity. And I'm going to just now highlight 3 cases very quickly. The first came out in New England Journal of Medicine a few weeks ago by Choi et al. Studied a 45-year-old with antiphospholipid syndrome who was chronically infected for 154 days before the individual sadly passed, had been treated with remdesivir twice, with a Regeneron cocktail once, varying levels of viral titers throughout the infection. And they sequenced the genomes and they found that this population had diversified considerably relative to other viruses from the same region, which was Massachusetts. And they zoomed in on the genomes of these viruses and they found lots of mutations in the critical spike protein, which is where the rubber of the virus sort of hits the road of our host cells. They found 8 different spike mutations co-occurring during this infection, including this N501Y mutation that is another defining feature of the B117 lineage. In another case by Kemp Gupta et al. That came out around the same time, they studied another individual, a 70-plus-year-old individual who had lymphoma. He also had a long-term, ultimately fatal infection of more than 100 days, was treated with remdesivir and 2 doses of convalescent plasma. And this figure is actually showing sort of frequency diagrams of multiple coexisting lineages at different frequencies over time. And here's that radial phylogeny that I had showed earlier. This is specific to this case, and this is the case. The particular patient of study here. Here's another individual who was chronically infected. They sample again. You see these long branch lengths, and in this individual, they identified 6 different coexisting spike mutations, including the 2 amino acid deletion that I already showed earlier as a marker of B117. And then finally, most recently, a group from Pitt here, led by Kevin McCarthy and Paul Dupre, Gadi Haidar and colleagues, forgive me, I'm not mentioning all of you, I know who you are, had characterized convergent evolution of small deletions in the spike protein and other regions of the viral genome that had occurred repeatedly and were evolutionarily successful. And so these red branches are all derivatives of successful lineages that all have these particular mutations. And this one right here, again, is that 2 amino acid deletion and spike. So to summarize these cases, the way I see it is that chronic infections are basically provide a forecast in what could be selected given sufficient mutation supply. So just as looking into space is like time travel into the past, looking into chronic infections is like peering into the future. Remember, chronic infections are usually dead ends and fail to transmit. And if you're interested in just this last few slides, I really encourage you listen to the Radio Lab podcast called COVID Crystal Ball. It really explained this work better than I ever could. And this is what I do. But unfortunately, one of these mutations did successfully establish a lineage, and that's B117. And so B117 evolves almost certainly from a chronic infection. It sort of burst on the scene with a whole suite of new mutations, 23 mutations, including many in spike, including these ones that I've already mentioned, another spike mutation at P681, 681 P to H, another small deletion, and then another substitution. And I think the important point is when B117 evolved, it invaded and continues to invade. It's not invading the sort of the neutral cloud of variants that are just slightly different from the Wuhan 1 epidemic. It's invading actually an adaptive background containing, for instance, already the D614G mutations. And so that brings us to the fall. Beneficial mutations for viral transmission and or immune evasion are no longer limiting. So we're no longer limited then by the challenge of asking if one particular lineage relative to everything else has a selective advantage. And that's called phylodynamics. Phylodynamics is really hard, where you're basically asking questions about the structure of the phylogeny and its relative rate of spread compared to everything else, and then also controlling for complicated issues like demography, sampling bias, and so on. So that's where we were before. But once population sizes get bigger, we can basically ask a more simple population genetic approach, and we can ask if particular individual mutations or combinations of mutations are under selection. And one of the clearest signals of an adaptation by natural selection from population genetics is site-specific convergent evolution. And more specifically, we ask, are mutations evolving independently in different lineages and becoming more frequent? So one challenge of phylodynamics is that it's really based on sort of an n=1 comparison relative to the entire backdrop. Population genetics is a more powerful approach in this case because n equals many. Okay, so one reason in addition why we can take this population genetic approach is a huge increase in genome surveillance, led primarily by wealthy countries with major public health infrastructures like the United Kingdom, to some extent Australia, who has contributed a number of sequences, many other countries with robust public health infrastructure. And as of as of a month ago, we were ranked about 40th as a country in the nation in genome surveillance. We're moving up quickly thanks to sustained investments from the new administration and from the CDC becoming strategic and targeted and also unbiased surveillance. And so this expanded genome surveillance and importantly, rapid data sharing makes inferring these adaptations easier. And so I want to just quickly state I've got a financial conflict of interest. As Daria mentioned, the Microbial Genome Sequencing Center was born in my lab, but we spun it off as an independent company here that Dan Snyder, who's in this picture with me, is leading and providing genome sequencing for lots of people, including folks who are interested in COVID sequencing. And I posted this tweet as a timestamp. We kind of put out our shingle that we were ready to do viral genome sequencing back in April. And unfortunately, the two major people to take us up were only two. They were the Harrison Lab here at the University of Pittsburgh, who used a great deal of kind of, coins under the cover, under the couch cushions and discretionary money to invest in sequencing some of our viral population here around the region. And then Jeremy Camille, who's really been the hero for me in getting me involved in sort of a deep dive in viral genome sequencing from LSU in Shreveport. He sequenced a good chunk of all viruses from Louisiana. And frankly, the Harrison Lab with us has also sequenced a decent chunk from the state of Pennsylvania. It doesn't speak as well for our local efforts just yet. In any case, these data have been amazingly valuable. And one thing that we're really committed to at MIGS and working with other colleagues is rapid data sharing. And so I want to give you a quick story about that rapid data sharing. This is an email that I got from Jeremy. I'll say this is really developed in a full-fledged collaboration independent of the company. He sent on January 27th, 2021. I think I should have looked for the timestamp on the email. I bet it was 11 or 12 at night. Preliminary analyses suggest there may be a few interesting variants in the latest 367 sequences you just finished and uploaded to Box. I'm seeing ones with spike P681H, Q677H, and Q677P alongside unique constellations of other clade or lineage-defining polymorphisms. And so this is actually a snapshot of the data that he plugged into their own local build of GISAID data. He then wrote, my suspicion is that there could be just as well USA homegrown variants could be competing fine against the B117, perhaps explaining why it hasn't begun to take over yet. Well, I mean, that sort of sunk in that night, and I thought about it, and the next day, I sort of took a snapshot, and I said, "Oh my gosh, this is actually a thing." And what we noticed is that not just our own little sample, which had clusters of the 677P variant and some of these 677H variants, but those variants were collectively increasing mostly in the United States. So P is only found in the United States in this snapshot. And the H variants are more common in the United States than they are in the world, but both seem to be going up. And one reason that we were able to make this inference is that another group at exactly the same time was making the same discovery in their own sampling sequencing. So Daryl Dahman and Daryl Dinwiddie, the double Ds from New Mexico, also had been doing a lot of yeoman's work to sequence viruses from their region. And they had posted their data right away. And what we noticed are these clusters of viruses in Louisiana and New Mexico, and then these other variants that were sampled from around the way. And I just want to show you how critical timely data sharing is. So this was our snapshot of the P mutations on February 3rd, so only a few days later from our initial discovery. And here's where we are on March 16th, just a few days ago. There's vast more increasing prevalence. And you'll note the big difference is that Texas uploaded their data as well as some of the neighboring states. So the cases have gone up fourfold in this time frame. Around the same time, I thought, well, geez, looks like we've got maybe multiple lineages here. I don't have the chops to sort this out in a timely fashion. I think we've discovered something. So I reached out to Emma Hodcroft, who's one of the real geniuses behind NextStrain, who works with in Bern in Switzerland. And just over the next really week, we put together this preprint that's now under peer review. It posted on medRxiv on Valentine's Day. And together, we discovered at least 7 new variant lineages arising in the United States carrying mutations at spike 677. We named these for regional birds instead of the states where they were first detected. We thought that was a way to avoid the problem of of shaming places for doing adequate viral surveillance by naming their lineages after those states. And then over the next couple of days, we also began to collaborate or sort of build on existing collaborations with the Camille Lab with groups from other institutions, notably Mount Sinai and Ben-Hur Lee's lab, start to dig into what is going on at spike 677. Beyond the fact that it was 4 residues from this polybasic or furin cleavage site that is critical for entry. So just a snapshot, these are the virus lineages named after the birds. So a major, to say the major reason why our story sort of gathered as much media attention as it did was because of this strategy for scientific communication and naming them after birds. It's that strategy has faced some significant backlash since, both sort of snarky comments and actually a significant and well-reasoned appeal from ornithologists, because the birds don't need to be shamed either. In general, though, I think that these naming strategies are important because we wanted ourselves to just distinguish these lineages. Robin 1 and Robin 2, for example, we knew were from the upper Midwest. We knew that Pelican was from the Louisiana area, and so on. So just to update you, these lineages continue to grow in frequency. And so this is the fold increase of the 677 lineages just over the last 6 weeks. And they're reaching anywhere from the P lineage from 0.1 to 1% global lineage, but the H lineages are reaching 5%. So what's going on at position 677? In truth, we still don't know. The major reason is, we think, is that it's quite close to this polybasic cleavage site, which is in the S1, S2 boundary of the spike protein. And that's very consistent with its potential functional relevance during cell entry. And the fact that this mutation repeatedly evolves, we think, is prima facie evidence of an adaptation, but how exactly it improves transmissibility or spread, we're not clear. We're delighted to be working with the Lee lab, who is building these mutants in engineered VSV cell lines with different reporter proteins, so we can actually do head-to-head competitions of the mutant and wild-type viruses, which is sort of back to my original beginnings in this field, where I was doing competitions in Rich Lenski's lab between an evolved mutant and a wild-type. Another point that Stephen Goldstein highlighted our attention to is that actually, while the glutamine Q is conserved in the original SARS-CoV-2 and in bat coronaviruses, it's notable that the human endemic or human at least adapted coronaviruses, HKU1 and MERS, have prolines there, just like the proline that we've seen evolving. So it appears that there's something potentially adaptive in putting in that protein, that proline, which might be altering the structural rigidity of that region to alter the pattern of protease cleavage. And something else that Jeremy has continued to remind me, it's not just furin that acts on this cleavage site. It's actually, there's a spectrum of proteases that could cleave here, and it's possible these changes are mediating that cleavage. And one other point that is really brand new, we're now just noticing that lineages at 675, Q675H and Q675P, again, H and P from an ancestral Q, are starting to spread in their backdrop. And so these changes from glutamine to histidine and proline in this particular region are strong candidates for adaptations. And so I'm just going to finish this talk to really point on this, that convergence we're finding is more the rule than the exception in this pandemic. And this is, I think, highlighted beautifully, or at least visualized beautifully, by this site developed by Emma Hodcroft called Covariance. See also the outbreak.info site from the Anderson lab at Scripps, showing very similar things. And so I want to point out, I think the similarities of these variants really deserve our attention more than their diversity. So the first is this 69 to 70 amino acid deletion. That's since been shown to be solely responsible for increased infectivity as part of those studies of chronic infections. The very well-known and fearful, fear-inducing anyway, E484K mutation that's responsible for a decreased susceptibility to neutralizing antibodies. That's evolved repeatedly in multiple independent genetic lineages. The N501Y mutation, that alone can increase transmission. That's work from Weaver and Menachery and others. These collection of mutations also convergently evolving. And also we're finding mutations either at 681 or at 677. And we have a kind of flimsy but tempting hypothesis that since we don't see these mutations co-occurring on the same lineage, we wonder if that either one of them is sufficient for the local adaptation to alter potentially protease activity. Okay, so I came up with a metaphor to understand how all of these coronavirus variants are assembling, and I hope you like it. It may date me, and that metaphor is one of Tetris. Okay, and so what I'm showing you here are some animations of mutations in spike. All of these are spike mutations, and the point is that you can put these different mutations in different combinations, and those combinations fit together in multiple different ways. You don't necessarily need all of them, but you need some of them, and each lineage is acquiring them independently. Thankfully, we don't see much evidence yet that recombination is happening within the novel coronavirus in a way that has given rise to new variants. However, again, the UK leading the way with their extensive genomics and histories have now recently shown that there have been some instances of recombination even within the B117 lineage. So we think that these lineages are converging upon similar combinations of mutations. And actually, we probably should be following the mutations or the mutation combinations more, say, than the B.1.1, whatever lineage names. And so what does this convergence mean for the future of the virus? I sort of said maybe they're starting to run out of some solutions. That's probably an overstatement. But I want to back this up with some data. This is actually data from Marcus Dillon and me from several years ago. What we did is we collected mutations and measured their fitness effects from bacterial lines that were evolved at very low population sizes, similar to the small population sizes experienced by SARS-CoV-2. And what we found is that most mutations, as predicted, are neutral or deleterious. Some mutations were very fit. And so what I'm suggesting is that the very fit mutations like N501Y, Delta 6970, E484K, it seems that those are likely scarce. And overall, then and then moderately fit mutations, for example, the ones we've discovered, which are more common, those might be additional adaptations. And they might arise in different sets of not completely overlapping combinations. So overall, I'm offering the hypothesis that the repeated convergent evolution among SARS-CoV-2 lineages suggests that the virus may be running out of these very big benefit mutations. Okay, so to begin to conclude, many have argued, many who think more about this and more eloquently than me, that this pandemic is rewriting our textbooks. And I'm showing you a shot of one of Carl Zimmer's textbooks on evolution. And I'd say it's not exactly true. It's not rewriting our textbooks on evolution. It is providing even better examples of evolution in real time and examples of convergence that many people, hopefully students, can appreciate and want to know more about. So you've heard about convergent evolution, and the classic example, for instance, is the evolution of wing flight from birds and bats. Perhaps the N501Y and N501T variants that convergently evolve in different lineages will become the next example, but the key messages and mechanisms remain the same. So to conclude, we know that SARS-CoV-2 evolved from a bat coronavirus that can readily recombine. We know now that viral epidemics mostly evolve by random forces when controlled. And I think we've known that for decades, frankly. And the virologists who've been studying this in for other types of viruses are seeing their discoveries highlighted. But likewise, when cases spike, so does the force of natural selection on potential adaptations. So rather, random drift becomes overwhelmed by the power of natural selection when cases reach the millions or tens of millions. With these unprecedented infections, with increased genome surveillance, with heroic data sharing, convergent evolution has pinpointed viral adaptations, and it focuses our attention on how those variants work and how they may threaten us. Evolution is basically telling us where to look, what experiments to prioritize, and what functional studies to follow up on, and what vaccine boosters we'll need in the future. And finally, I'll again reiterate my hypothesis that this convergent evolution offers some hope. So if we can constrain this pandemic and limit new cases, I think that the sampling of new genetic variation is going to be will be limited, and that will ultimately weaken selection with the big benefit mutations already underplay in the variants of concern that you've heard about. This has been a massive team effort. I really want to thank the folks in CBAM, Eric Wright, Lee Harrison, Jane Marsh, and many others in the Harrison Group in our lab, Dan Snyder, the hero for microbial genome sequencing, Jeremy Camille and John Venturi, who have basically moved heaven and earth to shake money loose to sequence as many viruses they possibly can from Louisiana and surrounding regions and really grow our contributions to the understanding of the United States epidemic. Emma Hodcroft at the University of Bern and of Team NextStrain, the double Ds from New Mexico Health Sciences, who've basically done the same, done lots of unfunded or underfunded sequencing, Ben Hurley and Sope Oguntuyu from Icahn School of Medicine for the next chapters. And I really want to thank COVID Science Twitter, super evolutionary virologist colleagues like Adam Loring, who continue to post wisdom there that helps set my frame, the Team NextStrain, the COVID sequencing effort in the United Kingdom, efforts from the Departments of Public Health in Louisiana, New Mexico, and Wyoming, Stephen Goldstein and the University of Utah, and you for sticking with me throughout this. I appreciate it. Thanks very much.
Thanks, Vaughn. That was amazing. I will take us through some of the questions posted in the chat. We'll get through as many as we can before the hour is up. Going back to the paper that you presented talking about bottlenecks and quantifying bottlenecks. So Anne Carbunas was asking about how the bottleneck size was calculated in those studies. And then Matt Nicotra was also wondering for us non-virologists, how are transmission bottlenecks and infectious dose related to one another?
Yeah, okay. So I'll take Matt's first. I think there's been a huge amount of discussion about the relatedness between the sort of infectious dose and the severity of infection. So I'm going to take that part and set it aside because I don't know. But what I will do is say that if you have 1 million viruses and they are all genetically identical, then the effective population size of that genetically identical population of 1 million viruses is 1. So there is no genetic diversity and natural selection has nothing to act on. And so that leads me to Anne's point, which is how do we calculate effective population size? So in all populations, the evolutionary population size or effective population size is heavily weighted by the smallest sizes it goes through, which are the bottlenecks. And in viral infections, that is the transmission event. And because it seems that the number of viruses that actually gets transmitted any given COVID infection is on the order of 1 to 8 individual potentially distinct viruses. And also because there's very little variation really appreciably in most infections, the effective population size remains 1, even though there's maybe millions of viruses. Now, what causes the larger population size to get bigger is many, many independent lineages, basically expanding the multiplicity of those events. And that's what natural selection is seeing. It's actually acting on lineages or chains of infections rather than on individual infections. And so that's why the effective population sizes of COVID is probably, I mean, my gut is still on the order of hundreds, if not thousands at most, rather than the tens of millions that have of infections that have occurred.
Great. Okay, so another question that came up related to the studies you discussed involving immunosuppressed hosts. Don Burke asks, so why is replication in immunosuppressed hosts more likely to generate significant mutants than the same number of replication cycles among normal hosts, presumably multiple normal hosts? Is it because there isn't a bottleneck where the virus moves from host to host or some other reason?
Yeah, I love the question. I didn't get into that. I'd love to expand, but we've been studying evolution in immunosuppressed hosts for a long time, and particularly in the case of cystic fibrosis, but more recently we've been actually doing experiments in immunosuppressed animals. And what we find in just transmitting bacteria in immunosuppressed mice is that the bacterial populations basically surveys, acquires, and enables to persist larger numbers of mutations. And so that finding suggests that it is immunosuppression that is removing some of those variants from ever establishing a foothold. And maybe some of that is just simply the size of the population. Smaller populations are basically able to grab fewer randomly arising variants. But immunosuppressed patients also have the infection endure longer within them, which allows that process to continue for longer, basically increasing the net mutation supply. How the particular form of immunosuppression acts on the viral population is a really active and exciting area. I think that's really a huge area of opportunity from this pandemic and more generally in host microbe interactions. But I would say that it is both just the size of the population and the relative inability of the immune system to sort of prune certain lineages from emerging within the population.
Cool. Okay, there have been a couple of questions coming through the chat about recombination. Personally, I have become aware more recently about the idea of recombination in this virus. And so I'm wondering if you could sort of meditate on the concept of recombination for a minute. So Russell Schwartz, for example, asked, is recombination rare because we don't see co-infection of people with multiple strains to permit recombination? One thing that I think about is early on when the virus is largely clonal, right? Recombination may still happen, but when you recombine with a clone, we can't detect that when it happens. And then also, is there any reason to believe there's enough active spread in non-human hosts that could be facilitating recombination?
Oh boy. Love the questions. I think so. So all of the factors that you just mentioned are great explanations. First, at the outset, if any recombination happened, it was basically with yourself. So that has no effect. What's going on now in the pandemic? So how do we detect it? Well, we certainly don't detect it as it happens, but we can detect it after the fact, again, with these very detailed studies with whole genome, whole population sequencing. Of course, that's generally you do population sequencing with viral infections, right? So you're looking for basically evidence of minority variants in one or several individuals where lineages are actually traveling together, and then you're looking for evidence of that resolution. The other way you really do it, though, is just from a retrospective, asking if particular mutations tend to evolve together in pairs in a new lineage. That's unlikely if those two mutations are actually already present, say, on that are not already found in that lineage backdrop. So the simultaneous, for instance, co-occurrence of two mutations provides some statistical signal of recombination event. And so you just sort of apply that inference at scale, and you can find evidence that certain combinations of mutations appear to have moved from one branch to another, independent of their mostly random lineage differences before that. And then the third was about, is there any evidence of non-human hosts being sort of venues for recombination events? Not yet. Not yet.
Yeah, I suppose time will tell on that
Yep.
one. Okay, Tara Levin asks a great question about the hope that the major mutations have been sampled already. So it seems that it might be true that might be true on the original genetic background, but epistasis could certainly change the landscape going forward. So are you aware of any evidence for or against that idea?
There's a lot of good discussion on that possibility. And I mean, there's a little bit of evidence already for the cases where combinations of mutations have been made. So it appears that there is probably some interaction between the deletion mutation and the 501Y mutation, I believe. It appears that, I mean, I speculated that there may be some opposition between 681 and 677 mutations. You either need one or the other, but maybe both is not good. And I think Tara's big question is, what does the future look like? Could the landscape affecting the fitness of different mutations change? Yes, it can. What does conditions approaching herd immunity do for selection on these variants? Well, what it's probably going to do is strengthen selection for E44K. I mean, we sort of can see that already. That's how it happened. That seemed how it was selected already. And it seems to have evolved in a region that already had very high infection, a very high force of infection and sort of prior exposure. Yes, it's quite possible that mutations that are not as beneficial now become more beneficial as conditions change. And I think that's the major concern. The good news is that we have the tools and the surveillance to look for them. right? So, and I want to point out that none of these approaches are that new. They just need to be applied rigorously and with rapid data sharing.
Yeah, I totally agree. I was gonna wrap us up with one final question, which you sort of already answered, but more future looking. So what would it take to improve pathogen genome surveillance in the US and worldwide by the next pandemic, or maybe by the end of this pandemic? And who
Yeah,
drivers
thanks for thanks for that question, and something that many of us have talked about. The Harrison Lab and us and folks who are on this acknowledgement slide. So there's a big gap at the state and local levels with the expertise needed to analyze the data if they were able to get genome sequencing data. So that's an if. What would they do with it, and what would they act upon it? This is not that hard to do, but it does require some infrastructure and investment. So whole genome sequencing is a thing. It can be taught and used by really, we've shown that you can do it with high school students, so they can do it. Certainly public health officials can do it. My big concern right now is the current packages really support sequencing and not the sufficient infrastructure development required to build the national and even international surveillance network needed. And that requires parties in research facilities like ours, public health facilities at the state and local level, to basically be connected. And that requires people being trained. I think it's a wonderful time to be in this area. I can see that the CDC is offering fellowships to do some of that training. I hope that training goes up 100-fold in the next couple of years.
All right, I think that's a wonderful thought to end on. So I'll wrap up and say thanks again to everybody for your attendance. Thanks so much, Vaughn, for the fabulous talk. Please join us on April 20th at noon for our next CBAM seminar, where we'll hear from Dmitry Petrov from Stanford. Thanks again, everyone. Take care.